name is Lilei Zhang. I'm from Baylor College of Medicine. It's my great pleasure today to have Dr. Cricket Seidman with me. She's from Harvard Medical School, and this morning she gave an amazing talk on hypertrophic cardiomyopathy. Cricket?
You are incredibly kind. I am at Harvard Medical School, and I'm a practicing cardiologist at now MGB, which was formerly Mass General and Brigham Hospital, and we are now MGB1. I had the privilege of talking today about hypertrophic cardiomyopathy, and in particular, focusing about what we have learned from studying of human hearts. We've learned quite a bit that has expanded what we recognize from prior model systems, such as those in mice and iPS-derived cardiomyocytes. I think one of the really interesting things that's come from the study of human hearts has been an appreciation of the complexity of the sarcomere. The number of different protein-protein interactions in the thick filament is really quite extraordinary, and it's humbling to think about how a mutation in one of the two gene copies could do so many interactions that could be quite harmful to our patients. So one of the things we've elected to do is to try and look at if there are differences in clinical outcomes aligned with whether a patient has a mutation that is particularly interacting with another region of the sarcomere proteins. And that's been pretty informative. We've found that there are some interactions that are associated with earlier onset disease, higher rates of stroke, and progression to heart failure. I think that incorporating that into our clinical risk stratification would be really useful in terms of identifying those patients who really are at need at surveillance and interventions rather when feasible. The other thing that we've been really fascinated with has been an examination of human hearts from patients who do not have a pathogenic mutation, have all the clinical features of HCM, as you and I both know, but we can't find the cause. And when we studied the human heart from patients undergoing a myectomy, which is obtained, as you know, when the function of the heart is really quite good, what we found is that there are some shared similarities, but some remarkable differences. Most importantly, it appears that there's a lot more fibrotic remodeling in patients who do not have a pathogenic mutation compared to those who do have a sarcomere gene mutation. And that, to me, this speaks the need for much more precise therapies. We now have some myosin inhibitors that we can use and are effective, and I think they would be most effective in those who have a mutation, whereas what we really need are these antifibrotic therapies for the people who do not have those mutations.
This is very interesting. As we learn more about the nuances of people with sarcomeric mutation, without mutation, with the myosin interface
Yeah.
mutation, and outside of the interface mutation, what is your strategy of thinking about stratifying these patients and giving them targeted
Yeah.
therapies?
Well, I think right now, unfortunately, we don't have anything more targeted than a medicine that directly impacts the sarcomere. And there are two right now that are leading the pack, if you will. One is mavacamten and the other is aficamten. And they've shown great responses in a lot of patients. But for the patients who don't have those mutations, while they're being treated with these medications, I have the sense that we need to do better and really address some of the fibrotic mechanism. And as you know, we heard some very exciting stories this afternoon, in particular about being able to suppress fibrosis with CAR T cells from Jonathan Epstein's group. And those kind of approaches may have more value in preventing progression of disease in patients who don't carry those mutations.
Do you perceive we will continue to see hypertrophic cardiomyopathy as one entity, or we will specify more subcategories and treat them differently?
Well, I guess I'm a splitter rather than a lumper, and I would prefer that we have more precise therapies that are going to be effective based on disease mechanisms. The other group of hypertrophic patients we really haven't talked about at all today are those who have thin filament mutations. And some of those are quite devastating, in particular in troponin I, where there's quite a bit of fibrosis associated with clinical patients, as well as in the troponin T mutation. So being able to find strategies to address those would be really, really beneficial. I had the privilege of sitting with a trainee at our lunch sessions today, and they are working quite hard to look at molecules that will target the thin filament and compared to thick filament. So I think we have opportunity there, and the young people will lead the way, which will be great.
That's wonderful. One of the main challenges in studying adult onset genetic condition is there's reduced penetrance.
Yes.
And what is your approach for these patients with positive genetic mutation but does not have manifest phenotype yet?
Yes. Well, it's interesting because I think that clinically we have routinely relied on morphologic remodeling of the heart as the diagnostic criteria for hypertrophic cardiomyopathy, an increase in left ventricular wall thickness. But when we look at those individuals who carry a mutation in the context of a family and they don't yet have hypertrophy, we see hyperdynamic contraction. We see poor relaxation. This was work that was spearheaded many years ago by my colleague Carolyn Ho, and we continue to see in families that I take care of today. It's my belief that that implies that hypertrophy is a late consequence, and the earlier we can begin to realize the opportunity to intervene, either with medicines or potentially with gene therapies, that we may actually prevent the remodeling from ever occurring. And that would really be a phenomenal opportunity.
That's really exciting.
Yeah.
I'm sure as a physician scientist, you have a very rewarding career to taking care of the patients, identify the genetic mutation, discover mechanism of the disease, and ultimately develop therapy. Can you tell us if in your research career, whether you have learned to ask the right questions by directly taking care of them, or whether they have contributed to for you to be able to make the critical decision during your research?
Well, those are very important questions, and I don't know that I have a simple answer, but I will tell you that the patients know so much about their diseases, and if we take the time to listen to them, they will tell you what troubles them the most. And that, in my mind, is a very important clue about where we need to look for answers of mechanisms. When we recognize that there's hypertrophy, we also recognize there's hypercontractility in HCM. But people don't get sick because their heart contracts too well. They get sick because it doesn't relax. And our patients have told us that for years. They're short of breath with exercise. They tend to retain fluid. And then, of course, they develop atrial fibrillation. So those kinds of issues have driven our understanding of where the problem lies. It's not necessarily the hypertrophy, it's the pre-hypertrophic phenotypes. And for many years, that has set us on a quest to understand what is the mechanism by which these mutations not only perturb contractility, but relaxation. And I think that with the development of these drugs, one of the key findings that they have been so beneficial for patients with it has been to improve relaxation capacity of the heart. That holds the opportunity to bring those kinds of scenarios to other heart diseases such as HFpEF and the like. So maybe it'll expand beyond HCM.
What do you see as some of the most important questions for the field in the next 5 to 10 years?
Well, in the next 5 to 10 years, I would hope that we will move beyond treating heart disease, and in particular genetic heart diseases, to actually preventing it. When you know the root cause of a disease, a gene mutation, there's opportunity now to intervene to correct that mutation or to silence the one mutated gene copy. And there's huge progress being done in preclinical models and cells to advance those kinds of technologies. I believe that we will get there. There are still many barriers. One of the first things we have to do is be certain that we know how to appropriately manipulate DNA and target what we want and not have unintended consequences. And equally so for our patients to understand that those kinds of therapies are enormously impactful and they come with risks early on. We need to recognize them, discuss them, and have really good conversations with patients, with payers, and with the whole healthcare systems.
It's really exciting. Thank you so much.
No, thank you. It's been a pleasure.